Germ Theory of Disease and Koch's postulation. Germ theory of disease stated that specific microorganisms known as pathogens are the cause of specific disease. That means infectious disease is caused by pathogenic microorganisms or germs. This theory marked a significant shift from earlier beliefs that diseases were caused by supernatural forces or imbalances in bodily fluids. The germ theory laid the foundation for modern microbiology and medicine. Louis Pasteur and Robert Koch were the key contributors to the germ theory. Louis Pasteur demonstrated that microorganisms cause fermentation and spoilage, proving the presence of germs in the environment. Robert Koch established a systematic method to link specific microbes to specific diseases. In order to prove whether or not diseases are caused by microorganisms, Koch used mice as experimental animals. Using appropriate controls, Koch demonstrated that when a small amount of blood from a diseased mouse was injected into a healthy mouse, later disease, the first experimental disease, anthrax, developed quickly. That means Koch took blood from this second animal, injected it into another, and again he observed the symptoms of disease. Koch discovered that the anthrax bacteria could be grown in nutrient fluids outside the host, and even after many transfers in laboratory culture, the bacteria still caused the disease when incubated into healthy animals. On the basis of this experiment and others on the causative agent of tuberculosis, Koch formulated a set of rigorous criteria known as Koch's postulates for definitely linking specific microorganisms to specific disease. That means Robert Koch formulated a set of criteria to establish a causal relationship between microbe and a disease. This is known as Koch's postulates. The first postulate of Koch is the microorganism must be found in all organisms suffering from the disease but not in healthy individuals. The second postulation stated that the microorganism must be isolated from a diseased organism and grown in pure culture. The third postulate, the cultured microorganism should cause the same disease when introduced into a healthy susceptible host. The fourth postulate, the microorganism must be re-isolated from the experimentally infected host and identified as being identical to the original microbe. Let us see modes of disease transmission and ways of prevention. The main routes of disease transmission are the first one, droplet infection. This is the route in which airborne disease passes from infected host to healthy host. Example, common cold, pneumonia, and TB. The second route is drinking contaminated water. This is the mechanism in which waterborne disease pass to healthy host. The best example, cholera and typhoid fever. The third route is eating contaminated food. This is the way that foodborne disease pass to a healthy host. The best example is salmonellosis, typhoid fever, listeriosis, and botulism. The fourth route is direct contact. This is the route in which pathogens spread by direct contact with an infected person or contact with a surface carrying the organism. The best example is athlete's foot, ringworm, and trachoma. The fifth route is sexual intercourse. This is the way in which organisms infecting the sex organs can be passed from one sexual partner to another during sexual intercourse. Example, candidiasis, syphilis, AIDS, and gonorrhea. The sixth, blood-to-blood contact. This is another way in which pathogens pass from infected person to healthy person through donation of unscreened blood. Example, AIDS and hepatitis B. The seventh one is animal vectors. This is the way in which many diseases are spread through the bites of insects. Example, malaria and sleeping sickness. We can prevent disease by having the following techniques. The first one is by having personal hygiene. Second, through vaccination. Third, safe food and water practices. Fourth, avoiding infections via different methods. Fifth, environmental sanitation. Seventh, maintaining balanced diet, exercise regularly, and get adequate sleep through quarantine, monitor and control vectors. And lastly, promote health education and create awareness.